Hello guys, welcome to my YouTube channel. So today we are going to discuss one of the applications of synthesizing labeled DNA probe, and which is famously called as the colony hybridization. Okay, so if you have observed my previous lecture on the making synthesizing DNA probe by random primer method, so there I was talking about the application where a probe goes and hybridizes with its target DNA, and from that we can figure out that if this probe, which is known to us, is hybridizing to a target DNA, that means the complementary sequence of the probe is present within the target DNA. Okay, so in this way we can segregate and draw some conclusions. So this is one application. So you have an agar plate. Okay, and this is a transformation experiment. So I think many of you know the experiment of bacterial transformation. Okay, so if you don't know, just go and look at it. What is bacterial transformation? So I'll give you a brief idea. In bacterial transformation, you introduce a foreign plasmid vector or a DNA, okay, into a bacterial cell. And what happens, that plasmid vector is expected to contain some that is actually one recombinant DNA. Usually it's a recombinant DNA where it will have some recombinant traits, for example some antibiotic resistance genes within it. And when you allow to grow it on an antibiotic medium, the transformants will grow. Okay, that means those bacterial cells which have that recombinant DNA within their cell will grow, and those the bacterial cells who don't have that recombinant DNA within their cells would not grow on the antibiotic selection medium, because the antibiotic will kill those cells. So only transformants will grow and non-transformed cells will die. So this is the basic principle of bacterial transformation. So you have such a transformant plate. So this is a transformation or transformant plate, agar plate. So okay, just a second. This is a transformant plate, so that you don't get confused. So only these colonies that you see here, these are the transform cells. So after you see the growth of the transform cells, you transfer these cells to a matrix. So the matrix can be either nitrocellulose or a nylon membrane. Okay, nitrocellulose or it could be a nylon membrane. Now, just like blotting, okay, you have to press gently these membrane matrices over this colony, okay, and then the cells will get transferred from the plate to this matrix or the membrane. Okay, so just as you do in case of western blotting, southern blotting, like this. So after you get these cells on the membrane, then what you have to do is, these cells, you have to lyse these cells by various lysing agents. So some of the lysing agents which break the cell wall and all these things, and then you have to denature the DNA, so you get single stranded DNA. Okay, so and you have to make them deproteinized without any protein, and make them irreversibly bind to the matrix. So you have to make suitable procedures so that these single stranded DNA molecules get bound to the matrix. Okay, so we have several procedures such that the nucleic acids like DNA get bound to the matrix. So if you have read about southern blotting, then we have used several binding agents so that the DNA gets fixed to the matrix. And like formaldehyde fixing is one technique. So likewise, and we remove the unbound, wash away the unbound DNA. So here in this third step that you see, you have the matrix and you have only the bound DNA. That means those DNA which can bind to the matrix, which are bound to the matrix, and the unbound cells are washed away. And after this step, you give the radio level probe. Okay, so this probe is what? Radio level single stranded probe. And you allowed it to hybridize with this DNA. Okay, so you'll allow it to hybridize with this DNA. So here, as you can see, this is a hybridized picture. So the probe has gone and it has found a suitable sequence in the target DNA, and so it has hybridized with the target DNA. So it may so happen that, as you see here, they have shown 7 colonies. So it might not happen that within all the 7 colonies, there will be hybridization. Maybe out of 7, there are 5 colonies where you have found hybridization. Or it may so happen that within the 7 colonies, there is hybridization, but not to the same extent. Okay, because it depends on the sequence of the target DNA. So if the probe has the complementary sequence of the target DNA, then only the probe will go and bind to the target sequence, otherwise it won't, right? So within a colony, we will have many single stranded DNA, and that does not mean that every DNA will have the same sequence, same complementary sequence as a probe, okay, and not also to the same varying extent also, okay. So by this process you can easily identify, and this is the region of positive response you can get in the autoradiography. So what will we do after this? You can cells from this positive colony can be subcultured, because they may carry the desired plasmid cloned DNA construct. So after you visualize it by autoradiography, so after visualization, you know that suppose within this colony you are finding the radioactivity. Okay, so you know this. Okay, this is my desired um, position or desired DNA, and then you can go on and subculture and preserve it, or you can perform your experiments. Okay, so subculturing. So this method is one of the most general methods called as colony hybridization. So this method actually is now obsolete. People don't use much colony hybridization except some older labs. If you go for like cancer biology labs or any lab which works on cell signaling pathways, if they're old labs, they will use these techniques. Otherwise, we have some modified versions also, some kit-based methods also. So generally, it is not used nowadays. But colony hybridization was one of the most like common methods earlier. Okay, similar to like colony PCR.